everyone, this is Fiza from Group 316. Today's topic is steady state of linear system. An open system may be in a steady, though non-equilibrium state. In this state, the entropy production inside the system is exactly compensated for by the flow of the entropy to outside the world, so that the total entropy of the system is not changed. Bertalan Feen, one of the founders of the thermodynamics of open biological system, termed such a stationary state that the state of low equilibrium. It differs from the true equilibrium by many specific features. The stationary state of an open system is achieved if the system is subject to a constant that fixes the constant value of a certain set of generalized force, but the generalized force may be vary. The entropy of such a system does not attain a maximum and the dispersion function sigma is different from zero. Suppose that the system exchange matter and energy with the environment, but this mass remains unchanged. In the stationary state close to equilibrium, the entropy production sigma is a minimum. This is the Prigoni theorem. It is directly associated with Onsanger's theorem and is valid only within the limit of applicability of linear thermodynamics. This may be illustrated by a spectacular model. The progress of a chemical reaction may be visualized as follow of a liquid. If the system is closed, one that is, if the liquid neither flows into or nor emerges from the vessels, then the entire liquid will flow at a velocity determined by the tap apparatus from the upper vessel to the lower ones, and the state of the equilibrium will set in. The level of the liquid in the lower vessel will represent the extent of a reaction in such a state. If the system is an open flow through system, a certain level of the liquid will be set up in the upper and in the lower vessel, which will not respond to equilibrium. This level will depend on the steady state flow rate, that is, on the extent to which the tap is opened. The steady state response of a linear system is the behavior of the system after the transient response by delayed to zero. Characteristic of steady state response: time invariant. The response does not change with time. Constant amplitude. The amplitude of the response remained constant. Constant phase. The phase of the response remained constant. Types of steady state response: steady state sinusoidal response, response to a sinusoidal input; steady state DC response, response to a constant input. Conditions for steady state response: first, stability. The system must be asymptotically stable. Linearity. The system must be linear. Third, time invariance. The system must be time invariant. Analysis methods: frequency domain analysis using transfer function and frequency response; time domain analysis using different equation and Laplace transform; phasors analysis using phasors to represent sinusoidal signals. Steady state response of linear system: first order system, exponential decay to steady state; second order system, oscillatory or non-oscillatory decay to steady state; higher order system, combination of exponential and oscillatory decay. Applications: control system steady response in crucial for stability and accuracy. Signal processing. Steady state response affect filtered design and signal analysis. Power system. Steady state response in vital for power grid stability. Passive membrane transport. The consideration of membrane transport should naturally be started with thermodynamics, without reference to the molecular structure of the membrane and transport mechanism. Thermodynamics not so much explain physically phenomenon as recognized our knowledge, establishing the relationship between physical phenomena and the dependence on the parameters of system. In this particular case, the thermodynamics reveals the dependence of the transport fluxes on the averaged characteristics of solutions and the members. Non-equilibrium thermodynamics offers a physical sustained description of passive transport. It determines the kinetic characteristics of a membrane which can be measured. Permeability requires a study of non-equilibrium flux of the material. The transport dynamics is tied up with a property of the membrane. The experimental found linear dependence of flux on generalized force for a number of passive artificial and biological membrane impels the nearness of the coupled process under consideration to equilibrium. Living cells are characterized by a fact that the concentration of K plus ions inside the cells may be 10 to 20 times higher than the surrounding medium. For Na plus ions, there are observed gradient of the same order but opposite sign. Experiment with the labeled atom show that the ions of a cytoplasm are exchanged for the ions of the environment, that is the cell membrane, is permeable to the K plus and Na plus ions. Passive membrane transport is the movement of a molecule across cell membrane without the using of energy. Types of passive transport: diffusion, osmosis, facilitated diffusion, ion channels. Diffusion: the movement of particles from higher to lower concentration. Random thermally driven process. No energy required. Example: oxygen, carbon dioxide, nutrients. Osmosis: movement of water molecule across semipermeable membrane from higher to lower water concentration. Important for cell volume regulation. Example: water absorption in kidney, plant water uptake. Facilitated diffusion: assisted diffusion using carrier protein or channels. Increased transport rate and selectively. No energy required. Example: glucose transport, amino acid transport. Channels: protein forming pores for ion passage. Selective for specific ions. Example: sodium, potassium, calcium. Voltage gated or ligand gated. Example: nerve impulses, muscle contractions. Key factor influencing passive transport: concentration gradient, membrane permeability, surface area, temperature. Passive transport mechanism: simple diffusion, facilitated diffusion, channel mediated diffusion, endocytosis. Biological importance: nutrient uptake, waste removal, ion hemocytosis, cell signaling, maintenance of cellular environment. Regulations of passive transport: membrane fluidity, channel and transporter expression, hormonal regulation, pH and temperature. Disease related to passive transport: cystic fibrosis, ion channel dysfunction, diabetes, glucose transport defects, kidney diseases, osmotic imbalance, neurological disorders, ion channelopathies, transporter related diseases, diabetes mellitus type 2, impaired glucose transport contributes to insulin resistance, Hartnup disease, defective amino acid transport cause renal and internal issues, cystinuria, defective amino acid transport leads to kidney stones, osmosis related disease, nephrogenic diabetes insipidus, defective water channels, impaired kidney functions, renal tubular acidosis, impaired hydrogen ion transport affects acid and base balance, edema, excessive fluid retention due to impaired osmoregulation. Thank you.